All right, so hello guys, I'm Dylan Feldstein, and we're going to be talking about central dogma. So I want you guys to type in the chat, what do you think those three molecules are on the screen right now? You can see this structure right here, and then there's three structures. So just type in the chat, what do you think those are?
Dylan, I can monitor the chat.
Okay.
Yeah, everyone got it. It's DNA, RNA, protein.
Awesome, guys. Okay, so the steps of gene expression. So you guys got it correct. So it's DNA to RNA to protein. So the central dogma of molecular biology describes the flow of genetic material in a two-step process known as transcription and translation. So in transcription, a single template strand of DNA directs the synthesis of a complementary RNA molecule. So in translation, this RNA molecule is translated into a sequence of amino acids to build a polypeptide chain.
So the purpose of the central dogma in this case is DNA is basically the genetic material of organisms. And when DNA is transmitted from parents to offspring in children, it can determine several characteristics, including height, eye color, hair color, whether you have a widow's peak or not. You might have seen it in your biology textbooks if you had learned biology. And then the DNA molecule is divided into units called genes, which provide the instructions for a specific functional protein. For example, proteins and functional RNA molecules, et cetera, enzymes. And then functional products of most genes are proteins, in particular polypeptides, which form into different shapes, which form into proteins like other proteins. And then genes provide the instructions to synthesize polypeptides, which provide specific functions needed by the cell or the organism. Basically, in short, DNA is organized into genes, genes encode proteins, and proteins direct cell function.
Okay, so DNA and RNA structure. So as you can see in the photo to the left, DNA is double-stranded, whereas RNA is single-stranded generally. And as you can see here, the difference between the sugars, DNA has deoxyribose as the sugar, whereas RNA has ribose as the sugar. And as you can see, the only difference is this ribose has an alcohol atom here, and whereas this deoxyribose just has a hydrogen. So the bases used in DNA are thymine, cytosine, adenine, and guanine, whereas in RNA, instead of thymine, it's uracil. And they also have cytosine, adenine, and guanine. So DNA and RNA are composed of nucleotides. So each nucleotide has a sugar, a nitrogenous base, and a phosphate group. And as you can see to the right, adenine pairs with thymine, and there's actually two hydrogen bonds between this pair. Guanine pairs with cytosine, and this pair has three hydrogen bonds between them.
Okay, so essentially, because there are four different bases that DNA is made up of, and then these four bases are then separated into two categories almost. Okay, so adenine and guanine, or we just refer to them as A and G, they are what we call purines, and thymine and cytosine, T and C, they're all pyrimidines. And now I don't think you don't really have to know the exact what that actually means. Just know that there are two categories. And in RNA, we have to know that uracil actually replaces thymine. So in RNA, instead of T bonding to A, you have U bonding to A. And if you look to the right at this picture of the DNA structure, you can see how DNA is actually built up. Okay, there. What is happening? Can you guys hear that sound? Okay, I don't know. You can see DNA and that it's made up of a bunch of base pairs, and in the middle you can see there's this hydrogen bond pattern, and you can see that every time you have an A, it always hydrogen bonds to a T, and every time you have a C, it hydrogen bonds to a G, and just like that, the DNA is formed. DNA is made, and it's always like this.
So I have a question for you guys. You can just answer it in the chat. Which bond do you think is harder to break? The bond between guanine and cytosine, or the bond between adenine and thymine? Just type it in the chat. Okay, so the answer is the bond between guanine and cytosine is harder to break because it has 3 hydrogen bonds, whereas the bond between adenine and thymine, as you can see in the photo, only has 2 hydrogen bonds. Also, as you can see in this photo, DNA has directionality. It has a 5' end and a 3' end. So as you can see, it runs in sort of like an anti-parallel structure, because this side has the 5' end first and then the 3' end, whereas this side is sort of like turned around with the 3' end first and the 5' end. So this is why DNA is anti-parallel.
So going back to earlier, we were talking about the central dogma. And if you remember, in that process, we had 3 different types of molecules. First, we had DNA, and then we had RNA, and then we had proteins. So the first step of the central dogma is actually transcription, where we take DNA and make it into RNA. That's a very big important step. And like if you want to remember the step easier, how I like to do it is like, generally, if you think of transcription, right, you will think of like back then when they had books and they had no printers, so somebody would have to like manually write it over into a new book, right? And so DNA to RNA, we're essentially just rewriting the code, and it's just into a 1 molecule strand this time. No, a 1 strand molecule this time. And so you can see there's these pictures telling you how it works. Essentially, what happens is 1 strand of DNA, it serves as a template for the production of another RNA strand. And how this works is because A always matches to T, or in this case, A matches to U, and G always matches to C. When you take half of the DNA, you can easily make another RNA with the help of some extra enzymes, because basically it's already mapped out for the enzyme. And so this enzyme is called RNA polymerase, and it builds the RNA strand with some floating nucleotides in the air in the 5' to 3' direction. So it always moves in the same direction along the DNA strand.
So stage 1 of transcription is called initiation. This is where a protein called RNA polymerase binds to a promoter. Basically, that's a sequence of DNA upstream, basically before the transcribed region, which is basically what's going to be turned into a piece of RNA. And the RNA polymerase basically unzips the DNA, providing a template needed to synthesize an RNA molecule. So basically, what it does is it splits apart the 2 strands of DNA, making it easier to copy, and then it copies off like that strand of mRNA, which will be shown in the next slide.
So the second stage of transcription. Oh, sorry.
Oh, okay. So elongation, what happens in elongation is 1 strand of DNA acts as the template strand, as I said on the previous slide, and then RNA polymerase assembles a strand of messenger RNA, from 5' to 3'. That's the direction that it goes in. And the RNA transcript has the same information as the coding strand of DNA, but it consists of uracil, U, instead of thymine, T. So basically everything on the RNA has U instead of T. It has no T's at all, while the DNA still has its T's.
So the third stage of transcription is called termination. So sequences called terminators indicate that the RNA transcript is complete. So once these terminators are transcribed, these sequences result in the release of the RNA transcript from the RNA polymerase. So in eukaryotes, the RNA transcript is called pre-mRNA. So it still has to be modified to become mRNA. So some post-transcriptional modifications. So in eukaryotes, the pre-mRNA transcript must undergo a series of enzyme-regulated modifications before it can exit the nucleus, enter the cytoplasm, and go to a ribosome to be translated. So these modifications include the addition of a poly-A tail at the 3' end, the addition of a 5' GT at the 5' end, and also RNA splicing, which is the excision of introns, or you can basically refer to them as the non-coding areas, and splicing and retention of the exons, or the coding areas. So as you can see in this photo, at the 5' end is the 5' cap, and at the 3' end, there's a poly-A tail, which is basically a sequence of adenine nucleotides. And these modifications give the transcript stability and protect it from degradation by hydrolytic enzymes. So when the mRNA wants to leave the nucleus and enter the cytoplasm to go on a ribosome, it needs these modifications in order to not be degraded. So these modifications are very important. However, in prokaryotes, newly synthesized RNA transcripts can act as mRNAs right away and undergo translation. This is because prokaryotes don't have any membrane-bound organelles, such as a nuclear envelope, to separate the RNA transcript from ribosomes in the cytoplasm of the cell. So basically, in prokaryotes, transcription and translation occurs simultaneously. Okay, so step two is translation. So you want, essentially, now that you have an RNA molecule, you want to turn that into protein. And so in this step, translation, RNA is converted into protein. So essentially, the mRNA is decoded to synthesize a protein that consists of a specific chain of amino acids, which essentially encodes a polypeptide. So in mRNA, nucleotides, which consist of A, G, U, and T, provide the instructions for building a polypeptide. So groups of 3 nucleotides are actually called a codon. And there are 61 codons for amino acids, and each one specifies a specific amino acid. And there's about 20 amino acids found in proteins. So the AUG codon specifies methionine, which acts as the start codon to begin a polypeptide. So typically at the beginning of a polypeptide, AUG is the starting codon. So UAA, UAG, and UGA are all stop codons. These essentially tell the cell when the polypeptide is complete. So the collection of codons and amino acids is called the genetic code. It allows cells to decode an mRNA into a sequence of amino acids. So as you can see in this photo, mRNA codons specify a specific amino acid. So AUG specifies methionine, ACG, 309, and etc. So each one specifies its own amino acid.
So this is basically a codon chart. So basically, as Dylan said earlier, groups of 3 nucleotides are called codons, And basically each codon, each group of 3 nucleotides codes for a different protein, different amino acid, sorry. And there are 20 different kinds of amino acids. I'm not going to list all of them out right now. I don't remember all of them off the top of my head. But basically, yeah, there are 20 different amino acids, and each codon codes for a different amino acid. Two different codons can code for one amino acid. So basically, for example, PHE in the upper left-hand corner, both UUU and UUC can code for it. And also something to remember, I believe Dylan said this earlier, but AUG codes for methionine. It's the thing in red at the nearly the bottom left-hand corner. And that's basically the start codon. And then the stop codons, the 3 stop codons are UAA, UAG, and UGA.
And then there are transfer RNAs and ribosomes. So basically, the transfer RNAs or tRNAs, they're like molecular bridges. You guys have probably heard of them. They're basically molecular bridges. And then, so what they do is they connect the mRNA codons to the amino acids, as you can see on the picture, that they encode. And then one end on the tRNA carries the amino acid specified by the codon, and the other end has a sequence of 3 nucleotides called anticodons, as you can see on the picture right there, coding for CUG. And they're called anticodons because they connect to the codons, and the letters correspond to the codon. And then the ribosomes basically set up the site. So they are at the site of the translation where the polypeptides are built. They're made up of protein and ribosomal RNA. That's why they're called ribosomes. And 2 subunits, a large one and a small one are there. You can see on the graph. And then they provide the set slots APE sites where the tRNAs can find their codons, and they can provide their amino acids.
So you can think of these sites, these APE sites, as arrival, polypeptide, and exit. So here, like, you can't really see it right now, but the first site would be the A site, the arrival site. So the tRNA would come in and dock its polypeptides in the arrival site, and then it would transfer over to the polypeptide site. And this is where the polypeptide chain grows. And then at the end, when the polypeptide is complete, it will go into the exit site and be released from the ribosome.
All right, and then the first stage of translation is called initiation. So basically what happens is the ribosome assembles around the RNA, and then the tRNA carries the amino acid methionine and it's basically initiates. It begins to read the start codon AUG. And then the setup's called initiation complex. And you can see the methionine in the beginning, which is obviously the start codon. And then there is elongation, which is the second step of translation. And this is when the magic happens. This is when the amino acid chain gets longer. It continues to get longer. The mRNA is read 1 codon at a time, and then the amino acid corresponding to each codon is added to the polypeptide. So this is how the amino acid chain continues to grow. And then when a new codon is exposed, a matching tRNA binds to the codon, and then the existing polypeptide chain covalently bonds to amine, the end of the amino acid of the tRNA. And then the mRNA is shifted forward in the ribosome, exposing a new codon for reading. So that's basically how we can produce more amino acids. And then now is the termination of the cycle. So after we produce the amino acid chain, it stops to the either UAG, UAA, or UGA, the stop codon. And once a stop codon enters the A site of the ribosome, then a release factor binds to the complex. And then basically it separates the polypeptide chain from its tRNA in the P site and allows the polypeptide to exit the ribosome. Then the remaining components, including mRNA and ribosomal subunits, are released, and then basically the translation ends.
So after translation, what happened to the polypeptide chain? So after translation, the polypeptide chain may require some extra folding to be corrected into its 3D shape, undergo further processing such as the removal of amino acids, acetylation, methylation, or get shipped to a specific site in the cell, or even combined with other polypeptides. So essentially this acetylation, you may be wondering what that actually means. So basically these acetyl groups will come in and then bind to like the histone proteins in the DNA and essentially kind of loosen up the chromatin and kind of expand it out. So this increases the ability for the DNA to get transcribed. So it would increase the transcription of the gene. Methylation, on the other hand, is sort of the opposite. It sort of condenses the chromatin. So methylation is typically involved in epigenetics because it kind of like sort of silences genes. It sort of stops the transcription of the gene because it condenses the chromatin and then not be transcribed. So they're sort of opposites. So here's a video recap of Central Dogma.
Imagine we have a beautiful article with us. What if we want to make a copy of it? The best way would be to make use of a cast or a mold. Similar strategies are used by our cells. The most important task for a cell is to synthesize proteins based on the codes in DNA. But how are these codes read? What's the process that helps decode this language? And are proteins synthesized merely by the sequence of A, T, G, and C? The answer to all questions lies in the central dogma of life. But what exactly is the central dogma? Well, DNA makes copies of itself by the process of replication. Post-replication in every cell, the code of the DNA is cracked and transcribed into a messenger RNA. This mRNA is then read and the code is translated into proteins. This interesting concept is nothing but the central dogma. We know that the replication process is semi-conservative in nature. So now our focus will be on the two processes, namely transcription and translation. Let's begin with transcription first. As we can see here, the process in which mRNA is synthesized based on the parent DNA template is called transcription. The part or segment of DNA which encodes for a particular protein is called gene. So how are proteins made from the sequence of base pairs in the gene? Let's go in the reverse direction now. Proteins are a specific sequence of amino acids. So the various amino acids come together in a specific series to form a chain that makes up the proteins. Now, to get the amino acids in a series, we need a set of instructions. These are present in the form of mRNA. And where does the mRNA come from? It is synthesized from a special template. Can you guess what this template is? Give it a shot. That's right. The template is a gene. So a segment of the DNA that encodes for protein is copied first. The copy is in the form of mRNA. This process of making mRNA is called transcription. Now, can you tell me why this process is needed? Well, it's quite simple. If there is no transcription, then how will mRNA be synthesized? And if that's not done, then how will the proteins be made? Hence, transcription is vital. But it's not as easy as it seems. There are several enzymes and substeps involved in the process. We are referring to the steps like opening of the DNA double helix at a specific point, assembling the set of enzymes, gathering of nucleotides, and so on. After all these steps, an mRNA is formed. The mRNA is like an exact copy of the DNA sequence that codes the protein. The only difference between the two is that mRNA is single-stranded and the base thymine is replaced by uracil. Now that the mRNA is ready, it's all set to enter the cytoplasm. That is where the next step begins. It's the translation process. Let's have a look at this interesting process in the upcoming part. Imagine you're appearing for an exam, but the paper presented to you is in a language that you don't understand.
Oh, sorry about that.
Imagine we have a beautiful article with us. And you are prepared for the exam, but interpretation of the questions is a problem. What can be done? Some help from a translator would work wonders, right? So translation is the simplest way to solve this problem. Clearly, a wise move would be to decode the unrecognizable language and then attempt the exam. Now,
Sorry guys, so this video actually forgot to mention the post-transcriptional modifications. So remember, like, once the mRNA is sort of transcribed, it's not complete yet because it can't exit the nucleus. Otherwise, it would be degraded. So instead, it has to undergo a series of post-transcriptional modifications, which remember include adding the 5' cap, the poly-A tail, and also RNA splicing, getting rid of the introns, and then only having the exons, which are the coding sequences.
imagine this cell to be in a similar situation. Let's assume that this cell encounters a set of instructions for performing a task. But the instructions are written in a coded language. So all it needs to do is to translate this language and then perform the task. Any idea which process are we talking about? That's right, we are talking about the translation process. In continuation with the process of transcription from the previous part, let's now understand the next step of the central dogma of life. As the name suggests, translation means converting something into a simpler, understandable form. And what's getting translated here? That's correct, the code hidden in the mRNA molecule. The process of transcription helps in copying the code written on the DNA. That means it forms the mRNA. Now the mRNA comes out of the nucleus through the nuclear pore and enters the cytoplasm. Here, the process of translation begins. To begin with, let's find out the directionality of this mRNA molecule. This is the 5' end of the mRNA, and this is the 3' end. And how do we know this? Are we randomly stating this? No, it's not random. We can recognize these ends with the help of specific markers. The 3' end has a continuous series of the A nucleotides. This region is called the poly-A tail. The 5' end, on the other hand, has a cap of methyl group. Thus, this end is said to be methylated. But why is the directionality needed? Well, it's extremely important. The assembly that carries out transcription binds to the mRNA near the 5' end. So how does the assembly know that this is the 5' end? The easiest way to recognize the 5' end is by the presence of this methyl cap. This is how the assembly understands to bind near the 5' end and not to the poly A tail. Now, what exactly is this assembly that we're referring to? Well, that's the ribosome unit. 2 units of ribosomes, one small and one large, form an assembly that helps in the process. Ribosomes are a complex assembly which acts like a molecular machine for protein synthesis. Remember the small dots attached to the surface of the rough endoplasmic reticulum? We find them in the diagram of a typical cell. These are nothing but ribosomes. So the ribosomes are usually made up of 2 subunits, a smaller and a larger one. The smaller subunit binds to the mRNA at the initiation site. Once this is done, the larger subunit binds and completes the assembly. Now, the other type of RNA, called tRNA, steps in to get the remaining work done. At the end, a chain of amino acids is formed and released from the ribosomal unit. So doesn't this chain of amino acids look familiar? You got that right. It's our protein. The chain is further modified to achieve a proper configuration so that the protein becomes functional. This is how the translation process works. The code of mRNA is translated and the protein chain is synthesized out of it. The process of transcription and translation together are called as gene expression. The code of a gene is transcribed first and then translated into the form of proteins. There is an extremely important thing to note here. Whatever we've talked so far is all about the processes in eukaryotes. Such high level of organization is found only in eukaryotes. But we know what eukaryotes are, right? A cell with a well-defined nucleus and a highly organized system is a eukaryotic cell. On the other hand, cells which lack a proper membrane-bound nucleus and organization are called prokaryotic cells. Since they lack the proper membrane-bound organelles and nucleus, they do not undergo such complex and sophisticated processes. So the replication process in prokaryotes is slightly different. This was a brief idea about the transcription and translation process seen in the eukaryotes. For more interesting concepts in genetics, tune into our channel and don't forget to subscribe.
Okay, so now we have some practice worksheets that I want you guys to do. So if you see to the left, right, it says for the following examples, give the appropriate sequence of DNA, mRNA, and tRNA and/or polypeptide. Remember, a codon chart can only be used for decoding a strand of mRNA. So if you see, for example, 1, you have a specific sequence of DNA. And so then you have to convert it to mRNA and then amino acids. So you can use this chart, the codon chart to the right, to help you. And so, yeah, I want you guys to work on that for maybe 10 to 15 minutes. Okay, so awesome job on that worksheet, guys. We're going to go on to the next one. So practice worksheet part 2. So I want you guys to basically label the summary of protein synthesis diagram below using the following terms. So transcription, translation, DNA, mRNA, ribosome, tRNA, amino acid, polypeptide, nucleus, codon, and anticodon. So just take out a piece of paper and label it 1 through 11, and then just label each of the following parts of protein synthesis. So I'll give you guys around 10 to 15 minutes to do this too.
And don't worry if you weren't able to finish the first worksheet. We'll
Yeah,
go,
we can
we'll,
always go
we'll
back.
just go over it.
Yeah. Are you guys almost done or do you need more time? All right, let's go over the answers. So for part 1, so TAC for mRNA, it would be AUG. So essentially it's just complementary. So GCG would be CGC. GGA, and then AGG would be UCC. GGG would just be CCC. And then TGG would be ACC. And then for the amino acid, it corresponds to the mRNA. So you would look at AUG, you would go to the codon chart and find AUG, which is right here. And you can see it's MET. So you would write down MET. CGC, find that on the codon chart. And you get ARG. So GGG would be GYL, UCC would be SER, CCC would be PRO, and then ACC is THR. So how'd you guys do with the first one? You can just tell me in the chat. Great job, Olivia. Awesome, Victoria.
Very good.
Hanman, you want to go over the second one?
Sure, I can do that. All right, on the slide. Dylan, I need the second slide.
What happened?
I need part 2 slides, like the answers to part 2.
Oh, you can't see it?
No, I can't.
Huh. I can just go over it, it's fine. But yeah, so for example 2, TTC would be AAG, GAT would be CUA. Remember that instead of thymine, it's uracil. TAG would be AUC. ATG would be UAC. CCG would be GGC. And then AAG would be UUC. And then for the tRNA, it's complementary to the mRNA. So AAG would be UUC. CUA would be GAU. AUC would be UAG. UAC would be AUG. GGC would be CCG. And then UUC would be AAG. So then when you go to the amino acid, remember, you're looking at the mRNA, not the tRNA. So instead of looking at UUC, you look at AAG. Remember that. So using AAG, you go to the codon chart. So you go to the codon chart, you find AAG, and you get LYS. So you write that down. Same thing with CUA. You go to CUA, and you get LEU. So you do that for all of them. How'd you guys do with example 2? Nice, Olivia. Good job. Awesome, Ria. Yeah, so really the key things to remember is that mRNA is complementary to DNA. The amino acids corresponds to the mRNA, not the tRNA. And then tRNA is complementary to mRNA. So if you guys understand those concepts, you'll get all of these right.
Nice job, Victoria.
And so for the last one, it's CAT,
Oh,
GG,
gee.
ATG, GAT, CCC, GTA. And then for the mRNA, it's GUA, CCA, go CCA, UAC, CUA, GGG, CAU. And then for the tRNA, it's CAU, GGU, AUG, GAU, CCC, GUA. And then again, for the amino acids, looking at the mRNA, you go to GUA on the codon chart, and you would get VAL. Same thing with mRNA. Same thing with PRO. You go to CCA, find it on the codon chart, and you get PRO. So how did this one go?
CCA Pro.
Very well. All right.
Okay, great job, guys. Okay, let's go on to the answers for the second worksheet.
Dylan, I can do this one if you don't mind.
Yeah, sure. Yeah.
All right, so basically this is labeling a summary of protein synthesis, right? So most of this should be self-explanatory, honestly, but one is the tRNA molecule. You got to remember it's shaped like this weird clover thing. And then number two is DNA. That should be rather self-explanatory. Number three should be mRNA. That's like the single-stranded thing that should also be relatively self-explanatory. Number four is a process, transcription. That should, it has a little mRNA with the unzipped DNA, so yeah, that should be relatively simple as well. Number five is translation. That's where, the number is a little ambiguous, I know, but it's describing the entire, it's enveloping like the entire process of like tRNAs attaching onto like ribosomes and then creating amino acids. So yeah, that works. And then number six is a ribosome. That's where the tRNA attaches, and then that's where the amino acids are attaching to a polypeptide. Number seven is an amino acid. It's attached on top of the tRNA, and it is brought by the tRNA to the ribosome where it becomes a polypeptide, which is number 8. Number 9 is the nucleus. Basically, it's a line that represents the nucleus, basically. And then number 10 is a codon. Remember, a codon is 3 nucleotides, a group of 3 nucleotides. And then number 11 is an anticodon. You got to remember that's the opposite of a codon, and that's what is on the tRNA. How'd you guys do?